Hi, my name is Tatiana, and I will be covering Homeobox genes in my video today. Animals are incredibly diverse. Compare, for example, a centipede to a bird, or a mouse to a fly, and it's easy to see the differences. How has evolution produced such diversity? Well, as we know, all animals start as single-cell embryos, and when you take a closer look at that embryo, you can't exactly tell what kind of species it will become. As the embryo evolves, it will develop many kinds of cells, such as tissue cells, muscle cells, or neurons, which is a process known as cell differentiation. Each is optimized for its appropriate use in the body. Remarkably, the DNA inside all these cells is the same, and at the same time, there is a process of morphogenesis. What makes cells differ from one another is that different genes in the DNA are either turned on or off in each type of cell. Specifically, regulatory DNA determines whether and when the gene will be expressed or turned on in a particular type of cell. If you're a brain cell, for instance, you wouldn't want the genes encoding hemoglobin proteins. One important class of transcription factors is encoded by homeotic or Hox genes. Found in all animals, Hox genes act to regionalize the body along the embryo's anterior to posterior axis, aka they control the timing and the route of development in the embryo. These genes all cluster together on the genome and play a major role in the development of animal segments, with different Hox genes being developed in different segments, which will become important later. Hox genes control the identity of body parts, setting up the body plan of the animal. Like, here's where you put the tail, and here's where you put the head. However, Hox genes don't give instructions on how to actually create the head, legs, or tail. Other genes do that. They're sort of the head architects of the body. They create the master plan, but they don't do the actual constructing themselves. They just tell the constructing genes when and where to do their jobs and how much of it to do. No gene in your body does anything unless it's told what to do and how much to do it. But what actually activates that first regulatory gene? Nobody really knows just yet. Hox genes are a relatively new discovery and are pretty underdeveloped in terms of our knowledge, which is crazy considering they code for the morphology, otherwise known as the forms of animals. The term homeotic, or homeosis, was coined by William Bateson in 1894. These words derived from the Greek word for same, when he stated that these words would mean that something has been changed into the likeness of something else. Hox genes were discovered by Ed Lewis in 1978, and his discovery and research with fruit flies proved to be monumental in the field of developmental biology. To further understand how these genes work, we will need a model species to observe, and in this case, we'll focus on the fruit fly, the Drosophila melanogaster, as well as the sand flea, or beach hopper, Parhyella hawaiiensis. When you observe a fruit fly, you can observe a head and tail end, then three segments of the thorax, one with legs, the other with wings, and the last with a structure called a haltere, as well as segments along the abdomen. One of the most famous experiments with Drosophila conducted by Ed Lewis is the mutation of the ultrabithorax gene, which is often referred to as UBX. In this case, the number of segments stays the same, but the identity of those segments has been transformed, whereas here, in the mutated fruit fly, the third thoracic segment has been changed into the second, causing a repeat of wings in comparison to the wild type, with only one pair of wings. In this example of a mutation, the mutation takes place in the antennapedia gene. We can see in the wild type, the antennas appear normal, whereas in the mutant, the antennae have been replaced with legs, which proves to be a pretty drastic transformation of the body. Or even in this mutation, where the legs have been replaced with eyes. To best understand how these genes work in fruit flies, let's take a look at this chart. There are 8 segments we can observe, and going from the head to the tail, the labial gene is the most proximal and is then followed by proboscipedia, deformed, sex combs reduced, and antennapedia, These complete the first half of the fly's body. The latter half is defined by the ultrabithorax, as well as abdominal A and abdominal B genes. All of these segments correspond to segments on the fly and manage the expression of whatever that segment controls. How does this relate to other animals or even humans? Well, studies show that mice are incredibly similar to humans in terms of how our Hox genes begin to set up. These genes split up into 4 clusters along 4 different chromosomes. Hox A is located on chromosome 7 on the 7p 15.2 band. Hox B is located on chromosome 17 on the 17q 21.32 band. Hox C is located on chromosome 12 on the 12q 13.3 band. And Hox D is located on chromosome 2 on the 2q 31.1 band. As we can see in this comparison, the way Hox genes are set up in mice and humans is pretty much the same, which is incredible considering we look completely different. To understand how these genes work in practice, we can take a look at the role of the 3 most posterior genes in the crustacean we mentioned earlier, the parhyela, and how they work together. UBX, abdominal A, and abdominal B are known together as the bithorax complex. The rule that this complex must follow is that the segments that lose expression will take on the identity of the segment in front of it, and then that transformation moves posteriorly until it reaches the boundary of the next segment. This will make more sense when we practice it. Working based off of this rule, we can assume that when we knock out the abdominal A gene, the segments overlapping with the UBX gene will take on the role of the forward walking legs, and the segments overlapping with the abdominal B gene will take on the role of the anchor legs as seen in this image. If we were to knock out the abdominal B gene, we can assume that the segments overlapping with the abdominal A gene will take on the form of the reverse jumping legs. However, since there is nothing to express from A4 through A6, UBX will actually gain expression in those more posterior parts to essentially make up for what isn't being expressed. What would happen if we were to knock out both abdominal A and abdominal B? Well, if we are to follow this pattern, we can assume that the segments that express those genes will take on the role of UBX and express the forward walking legs, which is what indeed happens. But what would happen if we were to knock out all 3 genes? Well, since UBX expands to the T2 segment, the gene that is expressed in T1 would show prevalence along the rest of these segments from T2 to A6, and the gene that will perform that expression is antennapedia. So what we actually see in these mutant parhyela is maxillipeds on each segment from T2 backward. In theory, these mutations can and have occurred in humans, just not in a formal laboratory setting because that would bring up so many ethical issues. While Hox genes can have negative effects on human evolution, it has an incredible adaptive value for other animals. While both the child and the seals share mesomelic dysplasia, which is a disorder of bone growth and shortens the limbs, it actually aids the seal in swimming. Now that we have a good description of what exactly Hox genes are, what makes up the Hox genes, how they work in practice in the Drosophila and the Parhyela, and how Hox genes relate to humans, I hope you can get an insight into just how incredible these genes are and how much they do during development.